Good afternoon, everyone. Thank you for coming. I'm so excited. This is the first for me, the Schofield lecture. It's like a big day, and we're so happy to have Jean-Pierre Lavoie. So I'll introduce Jean-Pierre Lavoie after. I just want to acknowledge the fact that the University of Guelph resides on ancestral lands of the Atawadanan people and the treaty lands and territory of the Mississauga of the Credit. We recognize the significance of the dish with the one spoon covenant to this land and offer our respect to our Anishinaabe, Haudenosaunee, and Métis neighbors as we strive to strengthen our relationship with them. Today, this gathering place is home to many First Nations, Métis, and Inuit people, and acknowledging them reminds us of our important connection to this land where we learn and work. So I would like to present Priya Jain, the president of CBSA, who will present the Schofield Medal to our guest today, Jean-Pierre Lavoie. Thank you, Priya, for coming.
Thank you very much. Oh, thank you very much.
Okay, and now I will present Jean-Pierre Lavoie, and honestly, I need my cheat sheet because it's so long. I actually met Jean-Pierre Lavoie several years ago when I was in St. Hyacinthe doing my internship and residency, and this is a long list of achievements, so I have to read it for you. So Jean-Pierre Lavoie is coming from St. Hyacinthe. He's a full professor of equine internal medicine, director of the Equine Asthma Research Laboratory and the Equine Respiratory Tissue Bank at the University de Montreal. He's an associate professor in the Faculty of Medicine at McGill University in Montreal as well. So Jean-Pierre graduated in 1980 at the University de Montreal, worked in equine practice. In 1984, he enrolled in a residency program in large animal internal medicine at the University of California in Davis. In 1988, Jean-Pierre completed a postdoctoral fellowship in equine pneumology at the same institution. He became a diplomate of the American College of Veterinary Internal Medicine, large animal, in the same year. So Jean-Pierre has a faculty position in equine internal medicine at the University of Montreal, where he shared his time between the clinic and his research laboratory. He initiated actually the first residency program in the Department of Clinical Sciences. I didn't know you were the first one to open that. His research program is primarily interested in the study of asthmatic inflammation and remodeling and their treatments. And second page. So his research has been funded by several prestigious funding agencies, such as the Canadian Institute of Health Research, the Natural Sciences Engineering Research Council of Canada, the equine and pharmaceutical industry. Jean-Pierre also trained more than 80 graduate students and has published more than 180 peer-reviewed publications. Jean-Pierre is also the editor of the 5-Minute Veterinary Consult Equine and has given more than 300 lectures worldwide. Jean-Pierre won also the Pfizer Excellence in the Research Award in 2005, the Zoetis Excellence in the Research Award in 2011, and the Vetokinum Excellence in Training Graduate Students Award in 2020. So thank you very much for doing us the honor of inviting you and speaking for us. So with no further ado, I'll give my microphone to Jean-Pierre. Thank you very much.
Thank you. Well, thank you very much, Alice. And actually, you covered most of my presentation, so I can short it up. Well, it's a great pleasure for me, and it's really an honor to be here for the Schofield Memorial Lecture. As I was learning about Dr. Schofield, I was really humbled by his professional achievement, but probably even so or more by his social implication and his contribution to the pro-democracy movement in Korea. So I don't know many veterinarians that combine so much between social, political, and professional activity. So I had the great pleasure of becoming a grandfather recently, and due to unforeseen events, I had to spend quite a bit of time with my grandson the last few weeks, so there's a little parts here and there in my presentation, it's a little bit bumpy. I'll blame it to that. So I guess I'll give you a little bit of background and tell you how I managed to get where I am right now. And actually, I'll just go back here. I choose the title for this presentation because just to highlight the fact that my research program really developed to answer questions that arise in the clinic, and I didn't know what was happening. And it then evolved and changed based on opportunities that were arising to me, and also from collaboration with people that really had expertise to complement mine. And the results of our research program ended up being directly applicable to the horse. Of course, I work with horses, but some of it also could be applied to human health as well. So as a bit of a background, I decided to be an equine vet when I was a teenager because I figured out that it was probably the easiest way to work with horses while earning a living. Later on, I realized I really didn't know what the life of a vet was. And then I graduated from the University of Montreal a long time ago, and I was very lucky at the time. We had great teachers, great practitioners, good horse people, really good horsemanship. But very little research at the time going on. And that was the beginning of the veterinary specialties, 1980. So we just had the young faculty joining. Diane Blais, some of you might know, unfortunately passed away too young. Paul Blouin and André Vrins was an equine internist, and it's really André that gave me a taste for internal medicine. And my two co-residents ended up, co-interns, ended up doing residency program in the United States. So Hélène Bergeron went to do a residency in reproduction in Texas A&M, and she was the first woman to go in the men's prison to do reproduction. It was apparently a big thing in Texas. And Anne Vachon did a residency and a PhD in equine surgery. But I went on to, instead I went to started my own private practice. That's the way we were doing it at the time. And I kept working in the clinic a little bit to help here and there at the vet teaching hospital. And then I'm still not sure how that happened, but I received a call from Brad Smith that some of you might know because of the book, The Large Animal People, because Brad asked me if I wanted to go to do a residency at UC Davis, so I really never applied to it. And of course, I jumped on the opportunity, and I went to do a residency there where there are great clinician scientists. All of them, the faculty there, were all involved in clinic and doing research at the same time. And I was really fortunate to work with those people. And through my residency, my 3-year residency, I was involved in a number of research projects, and that's really what gave me the taste for research. It was clinically related project. The biggest one was on the use of bovine colostrum for newborn foal. But there's different other clinical topics that we study, and that's really what launched me and made me discover research. I've got a picture here of Sharon Spears at the bottom right, who was a co-resident with me, and she ended up being a faculty at UC Davis in equine internal medicine, and she was my windsurfing buddy, and it was probably among the nicest years of my life. Then I went on to do a postdoctoral fellowship in equine pneumology in John Pascal's lab, also at UC Davis, where I learned to measure lung function in horses. And we had, I don't know if you can see in the middle there, but there's Casey Kirperchuk, who was the research assistant in the lab, was a really good, an excellent lung physiologist, and really taught me a lot. But we had this fancy physiograph that at the time cost $60,000 US. So I can't imagine how much it translated into today's money in Canadian dollars. But it was very expensive because instead of having those little pencil that were moving up and down, there was a laser printer, and laser printer was just coming to the market. But for half a day we were spending in the clinic, we were spending doing recordings. We had to spend 3 weeks, both of us, deriving the parameters on those tracing from the laser printer. So it was very, very challenging and demanding, and a few papers came out of that. And then, as Elise mentioned, I became a diplomat of the American College of Veterinary Internal Medicine about 6 weeks after I became a father. So that was a little bit challenging. And then I went back to Montreal to accept a faculty position. And then I went up the ladder. And I was really hired to do clinic. So we do like we do here. So any cases related to internal medicine we deal with, whether it is exercise intolerance or falls or neurological problem. And we had the beginning in a heavy clinic load. It was 26 weeks in clinic and 26 weeks on emergency, and the clinic was quite busy. And this is one of the incentives to start the residency, because we were understaffed. We're just 2 faculty members to cover the full year, full emergency. And and we have, when we're on the clinic, same thing as it here, but we have the resident, the interns, the senior student, and sometimes international student as well, and you have to adjust your teaching to all those students at the same time as you have to deal with the horse and the client. So I love it, but it's quite demanding, and I did some formal lecture. So I'm more here to talk about the research part. So my research activity really can be divided into 4 axes. The first one is really clinical research, where it's usually a small intern project or a small resident project that would be based on cases we see in the clinic, discover, or if we report on a new disease or new features of a disease that's already known. And I like to think that some of those little interns project had an influence on some of those individuals. And one person that some of you might know, Steve Giguère. Steve did his internship with us at the University of Montreal, and I was directing his internship program. And we worked together on 3 cases, 3 foals with Rhodococcus equi, osteomyelitis, and he published that. And Steve went on to do a residency at New Bolton Center and came back here at the OVC to do a PhD degree with John Prescott. And Steve became a world leader really on Rhodococcus equi. And there's a few examples like that where I think an early exposure to research really had an impact later on. Unfortunately, Steve died from a tragic accident too young. So when I was hired at the University of Montreal, I realized probably how young and naive I was. So I did not negotiate a better salary. I negotiated a herd of horses and the space to study them. And, and I had not that much money to equip my laboratory, so I didn't have the $60,000 US to do it. So I wanted the equipment to measure lung function in horses. So I contacted Ed Robinson, which I had met at Davis. He came to visit John Pascoe when I was a postdoc there. And Ed Robinson is one of the father of equine lung physiology, at least in North America. And I asked him if he knew of a machine I could buy that would be accessible financially, and he said no. But there is a group at McGill University in Montreal, not too far from you, that I know they are world expert on lung physiology, so maybe you should contact them. And thanks to Ed, I contacted them, and that had a huge impact in the next parts of my life. Then at the Meekins Christie, I was put in contact with a young researcher that's Jason Bates, who's now one of the reference and one of the top 5 people in lung function in humans. And Jason just developed a small DOS software to measure lung function in rodents, and he accepted to try to adapt it to the horse. We thought it would be an easy deal. It's like many things, it took years before it worked, but when it worked, I had it in the lab for many years after that. And the other technique, see, research at the time were usually, a researcher usually would have one technique and pretty much do everything around the technique he had or she had. We decided to include also bronchoalveolar lavage cytology to the lab, and that's really a technique that was pioneered by Laurent Viel during his master degree. And around the world, we're all using the technique he has reported for the first time. And I'll acknowledge right away that Laurent in his PhD thesis really did a roadmap of what needs to be studied on equine asthma for the next probably 100 years. And a lot of the studies I've done, if I look retrospectively in his PhD thesis, he had mentioned about it. So my lab over the time changed, and now we study the horse from their lung physiology down to their molecular cell and molecular biology, and even to their nanovesicles, and I'll talk a little bit about that. So some of you are not familiar with the disease I'm doing my research on, so I'll just give you a little bit of background on that, so you can, on that condition, so you can follow. And on the name change as well. So we've known since antique time that some horses are stable and fed hay, they will develop a chronic lung disease. And the name has been changing based on the characteristic of this condition. It was recognized. So the first description is imputed to Aristotle. I read the translation of the text in Greek. You need a great part of imagination to figure out this is the disease I'm studying. So. Then it was reported that the name was proposed to be changed for equine emphysema because of this irregular appearance on the lung surface of affected horses at necropsy, and you can see it's irregular compared to a healthy horses. But it turns out that there is no emphysema in those lungs. It's really air trapping, air that gets sucked in and cannot be expelled. And then a group in Europe identified that neutrophils were the predominant cells in the airway, so suggested to use the term equine COPD, which some people still use, older practitioners. But in people, chronic obstructive pulmonary disease is due to inhalation of noxious gas, and it's really it's fairly irreversible. It's extremely difficult to control, unlike the horse disease that can be easily managed. So to highlight the fact that it was not a fixed airway obstruction, Ed Robinson and his group used the term recurrent airway obstruction, or RAO, a term that's also commonly used. And so all those names describe the same condition that's associated with domestication. It affects somewhere between 10 and 15% of adult horses kept in climates such as the one you have here. It's associated with clinical signs suggestive of lung disease, cough, nasal discharge, and so on. And it's incurable, and that's really driven a lot of my research. But it's manageable. Very few horses will die of this condition, despite they can be very miserable. But what really characterizes this condition is this period of labor breathing. I don't know if you can see there's quite a bit of light, but they are really they have dyspnea at rest. And they can lose a large amount of weight when they are in crisis for an extended period of time. And then another researcher used the term inflammatory airway disease, or IAD, to describe the milder form of the condition. So all those name changing all the time created a lot of confusion, and everybody was confused except perhaps a few specialists working in the field. So for that reason, I suggested to use the term equine asthma to describe this condition, as many of us were already using the term asthma when talking to clients and between ourselves. Because there are different diseases, but they have similarities as well. And a bit to my surprise, it was taken very readily. I wasn't sure how that would be taken. So if you have a horse with severe asthma and you treat them with corticosteroid, there will be a marked improvement, rapid improvement in their lung function. And so I've got many figures are like that, or a few of them, so I'll just describe it here. So on the y-axis, we've got the pulmonary resistance, and that's an indices of the airway obstruction. And we've got at baseline and after 1 week and 2 weeks of treatment in healthy horses in white and in horses with severe asthma in black. And you can see that after a week of treatment, usually we go back close to normal lung function in these horses. And corticosteroids are drugs, they are anti-inflammatory drugs. So all their effects is mediated to inflammation. But if you look at the percentage of neutrophil in the airways of those horses using bronchoalveolar lavage cytology, you will not normalize the airway neutrophilia. It persists. And that's actually an observation I did on clinical cases where we were getting those horses in the clinic, we were treating them, sending them home, and they were coming back to be evaluated again, and the horses were doing better, but the neutrophil were still present. So that was a question that occupied a lot of my research time. So I didn't know anything about cell biology. It was the beginning of molecular biology, so I went back to the Meekins Christie again. I did 2 6-month sabbatical, one in 95, one in year 2000. And that led me to be an associate member of this group and eventually an adjunct professor at McGill. And the interaction I got through with the researcher at McGill really helped me to develop my research program. So in the mid-1990s, it was the beginning of molecular biology. I don't know, I mean, some of you, I see that there's people my age or maybe a bit younger. But I felt at this point, 1995, that either I was getting into molecular biology or I would become a dinosaur very quickly. So I had to learn to hold the pipette and do the PCR myself. I mean, it was very difficult. I don't have very good hands, so that was challenging to me. And I work in Dr. Hamid's lab. Dr. Hamid was the new director of the Meekins Christie. He was recruited to bring cell and molecular biology to this group that was before really interested only in lung physiology. And Dr. Hamid, during his PhD in England, was one of the co-author of the first publication that reported on the polarization of T cells in asthma. They had reported that, I'm not going to go into detail, but you probably all know that T cells are lymphocytes and they are immune cells, and they really direct a lot of the inflammatory and the immune response. And Dr. Hamid, because of the work he's done, right, I think he's got 800-something publication, and he's got an H factor that's ridiculous. So Dr. Hamid used the technique that was implemented in human with probes designed for horses, and using in situ hybridization, we look at the lymphocytes expressing different cytokines, and we concluded that horse with severe asthma had also a Th2 type response, as it was reported in humans. And that was published in the Human Respiratory Journal with the highest impact factor. We had submitted to Nature. You know what's great about submitting to Nature? You know right away if it's rejected. So within 24 hours, they said, thank you very much. It's interesting, but not within the scope of this journal. But that was really puzzling because Th2 type response are typically associated with eosinophil, not neutrophil. And also, at the time, neutrophil were not involved in human asthma. That was the presence of neutrophil people realized neutrophil were playing a role in some form of asthma later on. So because it was so surprising and questionable at some point, I submitted a grant to CIHR saying there's nothing in the literature that could link Th2 type cytokines to neutrophil. This is what we've got. And the grant was rejected. And I learned a great deal from that. Probably the first one is if you apply to CIHR, it never goes the first round. You have to go at least 2 rounds, sometimes 3 before you're going to get funded. I also learned that from the comments I had from the committee was they liked the grant, but they didn't know about the horse at all as a model. So I was told you have to go and disseminate the good news that this could be a good model and try to get advocates for this. Another thing I learned is if it could be done in humans and rodent or a combination of both, they would never fund the research in horses. So it was a learning experience. So I turned it around and the following year submitted to INSERT, which funded the study. And through that, we did show actually that IL-4 could activate neutrophil. They could promote the recruitment of neutrophil in tissues. We did a number of studies that explored this, probably more than you would like to know about. But the question I had really was not that one. I went to the Meekins because of this dissociation between the lung function and the airway neutrophilia after treatment with corticosteroids. So Lauren Lecoq, a resident in equine internal medicine, was brave enough to embark in a small project with molecular biology. And her results really showed that if you take blood neutrophil and you incubate them with corticosteroids, you will have a suppression of both at the genomic and non-genomic level, and that's mediated by the receptor. So that was not answering our question. And Gaël put also this study, because some of you must know Gaël. Gaël is a veterinarian that did an internship in Montreal, a master degree in my lab, and they went to do a DVSC here in equine internal medicine. Now she's back in practice in France. And what Gaël showed was that the blood neutrophil are at least as sensitive to corticosteroids as the other leukocytes in the blood. And this is not a phenomenon that's specific to the horse. It was also true in humans. And without going through all the studies, we find out that it's the pulmonary neutrophil that are cortico-resistant, not blood neutrophil, that it affects some of the neutrophil function. So corticosteroids will inhibit some of the pulmonary neutrophil function, but not others. It is gene-specific, and it's the lung microenvironment that likely induces this cortico-resistance. And oxidative stress seems to play a role, as well as IL-17, which is a cytokine that's present in the lungs of these horses. Okay, now when you give a bronchodilator to horses, a potent bronchodilator, you have a marked improvement in lung function in the minutes that follows the drug administration, but you're not back to the level of lung function of healthy animals. And Laurent Viel, in his PhD thesis, had observed that there was some changes in the lung tissue that was present, and these changes were getting more pronounced as the disease was getting more severe, suggesting that there was a structure-function relationship between the changes in the lung and the lung function. Through a collaboration with Jim Martin, actually Jim Martin is a pneumologist at Meekins Christie Lab, and he's also a specialist in smooth muscle biology and asthma. And Jim was doing histomorphometric study in his lab, looking at the remodeling, the amount of smooth muscle surrounding the airways of rodents, rat model he developed. So using samples that I had kept from horses that died over the years, we find out that there was 2 or 3 times more smooth muscle surrounding the airways of horses with severe asthma than age-matched control. And importantly, if you look down here, so on the y-axis, you've got the amount of smooth muscle. On the x-axis, you've got the size of the airways, 0 being the smallest airway. And you can see in the red triangle that there was an increase in the airway smooth muscle at all levels of the airways, but it was much more pronounced in the smallest distal airway. So that was suggestive that as the smooth muscle increased in the airways, the disease might have progressed from the milder form to the severe form in some horses. So I tried again, I submitted again a grant to CIHR saying, okay, I cannot study the biology of the remodeling because it's a naturally occurring disease. So by the time I see them, the remodeling is already established. But perhaps we could use this model to study the reversibility of the smooth muscle. And in human asthma, especially in severe human asthma, the greatest changes are also in the periphery of the lungs, and they cannot study that in humans. It's too invasive. The means to access that part of the lab, the lung, is too invasive. So the grant was funded, and in order to study, to do those studies, we had to develop techniques to sequentially collect large pieces of lungs from the periphery of the same horse to do temporal studies. And we did that with a surgeon at the University of Montreal. We used thoracoscopic biopsy, and the size of the lung tissue we collect is about the size of the lung of a mouse, so it allows to do a lot of studies with that. And from this study, we first characterize it, and we find out that using our thoracoscopy biopsy, we show again that there is 2 or 3 times more smooth muscle surrounding the airways of asthmatic horses compared to age-matched control, that a 30-day exposure to an environment that get the horse symptomatic did not change the amount of smooth muscle, which made a lot of sense because otherwise they would die from it, from occlusion of the airways. We find that there is some deposition of collagen and also elastic fibers, and importantly, there is a really strong correlation between these changes and the lung function of horses, indicating that there is a strong structure-function relationship and that these changes have an effect on the horse's life. And we also look not only at the amount of smooth muscle, but also at the makeup of the smooth muscle. In this study, for instance, we showed that there is an increase in the fast-contracting myosin isoform when present that would contract much more rapidly. And and through another student that now a practitioner in the Guelph area, Esther Milares Ramirez, that some of you might know. She recently did a master's degree in the lab, and she also showed that there is increased angiogenesis in the airways of asthmatic horses, and that it was reversible. So it's really the reversibility we were interested in. So Mathilde Leclerc, during her PhD thesis, did a really difficult study in my view. She followed horses with asthma for 1 year, with severe asthma for 1 year, after horses were treated by keeping them in an environment when there is very little of the offending antigen. So we removed the hay and the bedding and so on. And we did the biopsies. We did a lot of testing, but we did the peripheral lung biopsy at 3 intervals. And she found out that there is no significant decrease in the amount of smooth muscle after 6 months, but after a year of removing all the antigen, there was a significant decrease in the amount of smooth muscle. She also found out that in the other cohort of horses, we kept them in the stable, treated them with inhaled corticosteroids, and we had also a significant decrease, but this time at 6 months. And another 6 months treatment did not further decrease the amount of smooth muscle, indicating, and there was twice as much airway smooth muscle than our age-matched control, and that was indicating to us that this remodeling might be irreversible. And I think on the equine side, the importance, and that also may be true in humans, is that we need to identify early on horses that will develop these changes to implement therapy before it's established, because we might never be able to correct it otherwise. And a bit to our surprise, the fibrosis of the airways were almost completely reversible, but again, after a year. So we went on to see, can we accelerate? Well, we had the 6 month standpoint. We didn't know if it was occurring at 1 month or at 3 months or so on. And we wanted also to test if we were adding a bronchodilator, could we increase the reversibility of the remodeling? And Michela Bedoni and other PhD students reported that whether the fluticasone is combined or not with a bronchodilator, there's again a 30% decrease in the amount of smooth muscle. And those are all different horses, by the way. So it seems to be something that's genetically quite fixed. And that's all that a bronchodilator did not really accelerate these changes. And it was present at 3 months, but not at 1 month. So for the practitioner here, that means that you need to have a minimum of 3 months of treatment before you can, with inhaled corticosteroids, before you can see a decrease in the amount of smooth muscle. Now those thoracoscopic biopsies are very invasive, and there is no way that would be done clinically in horses to assess the severity of their remodeling. So we wanted to know if we could do that with endobronchial biopsies, which are quite easy to collect in standing horses after doing bronchoalveolar lavage cytology. However, what we find out is when horses were in exacerbation of equine asthma, there was a decrease in the amount of smooth muscle in the biopsies, not an increase or a stable. And we suspected that this was due to an incomplete sampling of the airways because of a thickening of the epithelium and the lamina propria. So we undertook to study all the parameters that could potentially influence the quality of the biopsy, the type of forceps we used, the area where we biopsied them, if we used bronchodilators and so on. And we really concluded that's the problem. You cannot use the endobronchial biopsies in horses with severe asthma to determine the remodeling of the amount of smooth muscle in the central airways. So in order to study this, the remodeling of the central airways, we obtained funding from the Canadian Foundation for Innovation. And using it to test to see if when we do endobronchial ultrasound, can we non-invasively assess the remodeling of the smooth muscle. So the way it works, it's a high-frequency radial probe that you advance the working channel of an endoscope. You bring it into the central airways, and I cannot show it with this, but you can see that on the right. And then you advance, and when you're within the airway you want to sample, you distend, there's a little balloon around it, you distend it with saline, and the airway will be imaged. So if you look, you'll see it here. So if you look down, the balloon is being distended, the image is being formed. And by doing IBUS, or endobronchial ultrasound, on horses that had to be euthanized afterwards, we were able to do the histology and show a strong correlation between both. And using this technique, Dr. Bologna was able to show that by combining salmeterol with fluticasone, there was again a 30% decrease in the airway smooth muscle mass. And by the way, all those studies are done when we do the histomorphometric analysis of these analyses. The slides are coded, and everybody is blinded. So they don't know if they are treating horses at baseline or after treatment. And I think more importantly, the remodeling assessed by IBUS, endobronchial biopsy, was correlated with the remodeling, the amount of smooth muscle we were able to measure using histomorphometric on the thoracoscopic biopsy. So it shows that it is a non-invasive way to assess central airways. I'll go very quickly on that, but now we're also interested to see how neutrophil can contribute to the increase in airway smooth muscle mass. In this study, we showed that equine neutrophil secrete exosome. Exosome, I don't know if you're all familiar with it, but they are nanovesicles that pretty much every cells secrete in their environment. We were the first to report that neutrophil secrete exosome, but it's not that surprising. Pretty much all cells do it. And they have receptor on their surface, and they're so small they'll take the circulation and they will distribute it through the body. And we show that if you incubate exosome from neutrophils stimulated with LPS, the exosome would rapidly be home into the cytoplasm of the smooth muscle. They will go around the nuclei, and they will increase the smooth muscle proliferation. So that's a mean by which it could be done. It's an example. And very briefly, another part, the last axis of our research program really based on testing drug efficacy and trying to develop new therapy for equine asthma. So because the model works very well, it's quite easy for us to bring the herd and test the drug, do it blindly with controls and so on. And we did provide evidence based of efficacy and comparative efficacy between the drugs as well. We work with pharmaceutical companies to try to develop new class of drugs. So far, new class of drugs have failed to be of clinical interest. But we worked with Boehringer Ingelheim, and we ended up with a new product to treat equine asthma, which is an inhaled corticosteroids, clisonide. It was a learning experience for me to do that. Now I'm almost finished. I should be close to my 45 minutes now. So from all those studies, there was a ton of samples that were generated, blood, ball, tissue, and initially it was quite easy to know from which project they were coming, but we were at the point where we wanted to use those samples. We had to go through the logbook of all the students, and some were good, some were not so good. And it became very complicated, very difficult to use those samples. So again, through a collaboration and opportunity, I met a researcher, Dr. Eric Rousseau, at the Respiratory Health Network of Quebec, which is a Quebec network that brings together people, researchers interested in respiratory diseases, human asthma primarily. And Dr. Rousseau is a smooth muscle physiologist working in Sherbrooke, at the University of Sherbrooke in Quebec. But he's also a horse lover, and he writes articles in lay journals in his spare times talking about horses. And we started talking about horses around a beer, and it turns out that he was responsible for the development of the human asthma tissue bank. And he said, well, maybe we could, would that be of interest to you? And maybe we could develop that together. And we did that. Again, it was more work than we expected. Dorothy Binzel participated to one of the workshops where we gather a lot of some researchers determined what physiological data we should include in the bank and how should we keep the samples. And now from my own site, I've got samples from 200 well-characterized horses, 560 horse projects. There is more than 15,000 samples. And I've been doing a number of studies now without the manipulation of the horse, just from archived tissue. And I've been shipping them to people in Europe, Canada, North America, South America. So it's really nice to have it. And now the technique we had developed in horses with severe asthma, we now implement those techniques in the clinic in a prospective study in milder form of asthma. And doing the full evaluation, lung function, BAL, lung biopsy, and endobronchial ultrasound takes less than an hour. So it goes very smoothly. And with that, I will finish by acknowledging all the work that was done, that I presented today, was done by talented, motivated graduate students that brought new ideas as well and got the projects to evolve, the collaborators without which I would never have done those studies. And the granting agencies are really essential to get research going. And I will finish by saying that one of the challenges being a clinician scientist is to deal with crazy long days sometimes. So I would not have been able to maintain this activity without the help from my wonderful family. My wife, Sheila Laverty, some of you know, she's now a professor emeritus at the University of Montreal, an equine surgeon, a clinician scientist working with joint diseases. Our three kids, Sarah, Emily, and Patrick, and my grandson. And with that, I would like to thank you for your attention.